if we want to balance this, I said 2 protons plus a half a molecule of oxygen produces water. Okay. Now, what we've got in the diagram is, so far, we've taken glucose, we've completely oxidized it in glycolysis and pyruvate oxidation in the citric acid cycle. Now we're taking the energy that's left over in NADH or FADH2, which we haven't drawn in yet, and we're tapping into that energy. The NADH has these high energy electrons that flow through an electron transport chain all the way down to oxygen. They fall from a high energy spot to a much lower, more stable location. As they fall to oxygen, oxygen is the final acceptor of electrons. It is reduced to water. That's all happening here. Why is this so exciting? What happens essentially is we're using the energy in NADH or FADH2 and we're tapping into it. We're going to use this energy that's slowly released as the electrons fall down the electron transport chain to do some work. We're not going to quite make ATP yet, but we will soon, all right? So what's going to happen now with this electron transport chain is, as the electrons flow through the electron transport chain, energy is released. That energy is used to drive or to pump protons across the membrane. What? It turns out that these electron transport protein complexes also can carry or can pump hydrogen ions or protons across the membrane. And that takes energy to pump them against their concentration gradient. So what happens is, as these electrons are flowing down the electron transport chain, energy is released. That energy is used to pump protons across the inner membrane and into this space between the membranes. What we create then is an area in the mitochondrion that has a high concentration of protons, a high concentration of hydrogen ions. So I'm going to write that in. We have a high concentration of protons in the space between the membranes, much higher than down here in the matrix. I'm going to write down that we have a low concentration of protons down here. Okay, what have we just done? We've just used the energy in NADH and FADH2 as these electrons flow through the electron transport chain. That energy that's released slowly is used to pump protons across the membrane, creating a proton gradient across the membrane. We've got high proton concentration here in the space between the membranes and low proton concentration down here. We've created a gradient across this membrane. We're going to call this gradient something. We're going to call this gradient a proton motive force. Proton motive force. We use that word. I love that word force, because what does that imply? It's a source of energy. We've created a source of energy. We can use this proton gradient to do something else. We want to make ATP. We're going to use this proton gradient to make ATP. All right? We're just about there, folks. We haven't made it yet, but we're going to make a lot of ATP from this proton gradient. I just have a couple things I want to do yet. Now, remember, FADH2 can donate electrons also. FADH2 donates electrons to this complex called Roman numeral 2. It donates 2 electrons, and when it does, it loses those electrons. It's oxidized back to FAD. Those electrons then can flow through the electron transport chain as well, creating a bigger proton gradient across the membrane. So what's cool about the electron transport chain is it's essentially a way to tap into the energy in NADH and FADH2, tap into those high energy electrons, use those high energy electrons to release energy slowly. The fall of these high energy electrons down to oxygen is a way for this electron transport chain to use that energy to pump protons across the gradient, creating the proton motive force, right? We've got a concentration gradient across this membrane. That's going to be the way that we can make ATP. We're into our next step. Now we're going to make ATP. We've got this proton motive force, and now we have to use it to make ATP. We need to draw in one more important enzyme into our inner membrane. This enzyme is called ATP synthase, and I usually make a better drawing. Try to make a drawing of it that's sort of realistic, but I'm not real good at that, so don't panic. And I've got the wrong color again. I'm looking for my markers here. This always happens to me. Here it is. Here's purple. Okay. So what we've got essentially is a molecule that looks something like this. This molecule is an enzyme. It's called ATP synthase. We know it's an enzyme because it has that ASE ending, right? Okay, ATP synthase is going to be important here. What ATP synthase can do is it can allow protons to pass across the membrane. Protons can pass through ATP synthase and diffuse back across the membrane. And that's what they want to do. There's an electrical and chemical gradient that we just made. Protons are happy to move back across the membrane, okay? And ATP synthase is a kind of complex molecule with lots of different parts, but one of the things that it does is the protons sort of enter the ATP synthase, and after several of them enter the ATP synthase, it rotates, the part of the molecule rotates, and the protons can be released. The protons can diffuse across the membrane. Going back to where they came from, this is good. They're kind of wanting to diffuse across the membrane, if you will. But as they diffuse across the membrane, this triggers a change in the shape of our ATP synthase molecule. It triggers this rotor part of the molecule twists, and that causes a twist in the molecule, and that leads to a shape change in the catalytic center of the molecule, making this enzyme functional. So as protons move through ATP synthase, this enzyme can start doing its job. What is its job? Well, its job is to catalyze the making of ATP from ADP and inorganic phosphates to ATP. Guys, we did it.